Hi, my name is Sam. Thank you for judging my project today. The title of my project is Exploring IBD Genetic Upregulation in Alzheimer's Disease. Alzheimer's disease is a neurodegenerative disease which impairs cognitive functions such as memory, reasoning, and language. According to the Alzheimer's Association, during 2020 and 2021, it was the seventh leading cause of death in people aged 65 or older. By 2025, 7.2 million people are expected to have Alzheimer's disease who are over the age of 65. Unfortunately, there is no cure for this disease as of today. Scientists know some of the mechanisms which can lead to Alzheimer's disease, yet there is a large gap in the understanding of the causation of this disease. One risk factor, which is known as inflammatory bowel disease, or IBD. IBD has been shown to increase the risk of Alzheimer's disease sixfold. A possible explanation to why this happens lies in the immune system. It has been shown that immune system changes affect both of these diseases individually. What if the dysregulation of immune system cells is a shared cause of development for both diseases? While my prior science fair projects have focused on reaction time and factors such as emotional state on reaction time, I decided to focus on a specific neurological disease this year. I chose Alzheimer's because it was common, could lead to death, and had no cure. While I knew I couldn't work with patients with Alzheimer's disease directly, I could contribute to the understanding of the pathology of the disease. When reading about Alzheimer's disease, I came across an article stating the relationship between gastrointestinal diseases and Alzheimer's disease. I chose to stick with this relationship in hopes of providing new insights onto the underlying mechanisms of Alzheimer's. Before I move on, there are a few essential terms which you need to know. Gene set enrichment analysis, or GSEA, is a computational method used to identify statistically significant differences between designated classes of genes. T cells are a type of white blood cell which are under the category of lymphocytes. Sorry for stuttering. There are two main types, CD4 and CD8 T cells. Together, they are responsible for the marking, instigating a response for, and elimination of a harmful pathogen or virus. IBD, or inflammatory bowel disease, consists of two main diseases, Crohn's disease and ulcerative colitis. In this project, IBD will be explored through the lens of what it does, gastrointestinal inflammation, and what it does to the part of the body it affects, functional abnormality of the gastrointestinal tract. P-value is the probability of an observed result in an experiment. In this study, it will determine whether the results are significant or not. The purpose of my project is to determine if IBD pathway genes exhibit enrichment in Alzheimer's disease when compared to control. Specifically, this project hopes to determine if gastrointestinal inflammation pathway genes exhibit enrichment in Alzheimer's disease when compared to control, and if pathway genes associated with functional gastrointestinal abnormalities, including those with underlying immune system pathology, exhibit enrichment in Alzheimer's disease when compared to control. It is hypothesized that gastrointestinal inflammation pathway genes exhibit enrichment in Alzheimer's disease when compared to control due to the lack of regulatory T cells. It has been found that the cytokine TGF-beta is underrepresented in Alzheimer's disease. This cytokine is essential for creating regulatory T cells. Regulatory T cells are responsible for controlling and suppressing immune responses. If immune responses are left unmodified or unsupervised, then they can lead to chronic inflammation and inflammation in any part of the body, such as gastrointestinal inflammation. This theory is also backed by the correlation between IBD and Alzheimer's. It has been theorized in multiple studies that gastrointestinal inflammation directly influences the development of Alzheimer's disease when compared to directly influences Alzheimer's disease development. Due to this, it is highly likely that Alzheimer's disease that pathway genes in Alzheimer's for gastrointestinal inflammation will exhibit enrichment in Alzheimer's disease when compared to healthy controls. It's hypothesized that pathway genes which are associated with functional gastrointestinal abnormalities, including those with underlying immune system pathology, will exhibit enrichment in Alzheimer's disease when compared to control. This is believed to have been caused by gut microbiome. This is supported by the relationship between gut microbiome health and cholesterol level variations with their relationship with Alzheimer's disease. In a study on July 3rd, 2023, more than 10,000 individuals above the age of 71 had a 19% higher risk of developing Alzheimer's due to cholesterol level variations, due to gastrointestinal mutations being associated with a higher chance of Alzheimer's disease. Then it is highly likely that pathway genes which are associated with Alzheimer's, or pathway genes which are associated with functional gastrointestinal abnormalities, including those with underlying immune system pathology, will exhibit enrichment in Alzheimer's disease when compared to control. A brief rundown of my methods are going to the GSEA website, downloading GSEA, acquiring a transcriptome file, creating a CLS file, and downloading a GMT file, loading those into the program, setting the parameters such as how many permutations should be run, and then the GSEA is run. P-value and enrichment score were written down and analyzed. The averages, or enrichment score, was averaged and percent different. I'm sorry, let me redo that. The percent, or no. Percent difference. I'm so sorry. The enrichment score was averaged and percent differenced, and the p-value was written down. The results showed that gastrointestinal pathway genes associated with gastrointestinal inflammation had a non-significant trend towards downregulation in Alzheimer's disease when compared to control. It had the same amount of representation as in control, 10.2%. This graph right here shows that pathway genes associated with functional gastrointestinal abnormalities, including those with underlying immune system pathology, exhibited a non-significant trend towards upregulation in Alzheimer's disease when compared to control. However, it was shown that the control had 14.23% more enriched or representation than the pathway genes in Alzheimer's. The hypothesis which stated that gastrointestinal inflammation pathway genes will exhibit enrichment in Alzheimer's disease when compared to control is rejected. This is because they had the same representation, 10.2%, as control, and they had a non-significant trend towards downregulation. Additionally, the p-value was over the significance level of p is less than 0.05. While there was no correlation between gastrointestinal inflammation and Alzheimer's in this experiment, that is not to rule out that there might be a correlation of gastrointestinal inflammation and Alzheimer's. However, it is not shown in T-cell transcriptomes. However, it could be shown in, let's say, B-cell transcriptomes. Additionally, the sample size only had 127 genes. If there was a data set with maybe 10,000 or more genes, that could have proved that could have had a different result. The hypothesis which stated that pathway genes which are associated with functional gastrointestinal abnormalities, including those with underlying immune system pathology, will exhibit enrichment in patients with Alzheimer's disease when compared to control is rejected. This is because the p-value was over the significance level of p is less than 0.05. However, there was a trend towards upregulation with overall the average being, or with Alzheimer's, with the pathway genes, it having an 0.67 amount of enrichment in Alzheimer's disease. With the pathway genes in Alzheimer's disease had a positive enrichment score of 0.67. However, also, the pathway genes had 14.23% less representation in Alzheimer's disease than in control. While the results were not significant, the trend is suggestive of an association between the gastrointestinal system and Alzheimer's. It is possible that a larger data set may have yielded a p-value reaching the level of significance, although this set was greater than 1,000 genes. Again, if it was performed in the lens of a different transcriptomes besides T cells, then there might have been different results. In the future, this project could be modified by using B-cell transcriptomes instead of T-cell transcriptomes in a similar analysis as this study. Completed analyses with specific gastrointestinal diseases instead of data sets for general diseases, determining whether using specific gastrointestinal diseases associated with using B-cell transcriptomes. Complete an analysis with specific gastrointestinal diseases instead of data sets for general data sets of gastrointestinal inflammation or functional pathways, and determining whether genes which were considered as hallmarks for either gastrointestinal inflammation or a functional abnormality of the gastrointestinal tract would exhibit enrichment in Alzheimer's disease when compared to control, specifically those hallmark genes. Overall, this study could help. This information from this project adds to the underlying understanding of Alzheimer's disease pathology and connections to other systemic illness, including understanding the pathways associated with the gut-brain axis. Thank you so much for judging my project today. I'm so sorry I cannot be there. I am at a funeral at the moment you are seeing this. I hope you have a great day. I would shake your hand, but I'm not there, sadly. Have a good day.